Cellular respiration can be broken down into four major phases. First, glycolysis. Second, pyruvate oxidation. Third, the citric acid or Krebs cycle. And fourth, oxidative phosphorylation. The goal of cellular respiration is to oxidize as much energy out of glucose as possible.
The first phase is glycolysis, which means the breaking down of glucose, a 6-carbon sugar. Glycolysis is completed in aerobic, requiring oxygen, and anaerobic respiration, lacking oxygen. Glycolysis takes place in the cytoplasm. First, glucose, a 6-carbon sugar, with the help of 2 ATP, is broken down into 2 3-carbon sugars with phosphate groups attached.
Next, ADP comes and combines with the phosphate group on each 3-carbon structure to create 2 ATPs per each structure. The ATP in glycolysis is created by substrate level phosphorylation. This is because the ADP combines with the phosphate group connected to a substrate rather than a freestanding organic phosphate.
NAD+ also binds with the structure and removes electrons to form NADH. The final product of glycolysis is pyruvate. In the end, you have a net gain of 2 NADH, which will later be used in the ETC, 2 pyruvates, and 2 ATPs, because while you made 4, you initially used 2. Lastly, if oxygen is available in the mitochondria, pyruvate will enter to be oxidized.
At the end of glycolysis, we have 2 pyruvate molecules that both contain lots of removable energy, but cannot be used directly in the citric acid cycle. Therefore, when the pyruvate enters the mitochondrial matrix, it undergoes a series of reactions that removes CO2. Next, the now 2-carbon molecule is oxidized and the electrons lost are picked up by NAD+ to form NADH. The molecule is now acetyl. Next, a coenzyme A is attached to create acetyl-CoA, which acts as a fuel in the next steps of cellular respiration. In short, 2 carbons are released from the original 6 in glucose, 2 NADH are generated, and 2 acetyl-CoAs are formed. There is 2 of everything because there are 2 pyruvates undergoing this process. The citric acid cycle, also known as the Krebs cycle, takes place in the mitochondrial matrix. The cycle is a closed loop. It includes 8 major steps, and the last part of the pathway reforms the molecule used in the first step. First, the acetyl group of the acetyl-CoA joins the cycle by combining with a 4-carbon acceptor molecule, oxaloacetate, to form a 6-carbon molecule called citrate. Next, this citrate molecule loses 2 carbon molecules and NAD+ is reduced to NADH. The remaining 4-carbon molecule undergoes a series of additional reactions by multiple enzymes. First, making an ATP molecule by substrate level phosphorylation. Then, reducing the electron carrier FAD to FADH2. And finally, generating another NADH. This set of reactions regenerates the starting molecule, oxaloacetate, so the cycle can repeat. The cycle goes around twice for each initial glucose molecule, thus generating a net gain of 4 carbon dioxides, 2 ATP, 2 FADH2, and 6 NADH.
The last process is oxidative phosphorylation, which can be broken down into 2 subgroups, the electron transport chain and chemiosmosis. First, the electron carriers NADH and FADH2 bring hydrogen ions to the electron transport chain, a series of proteins embedded in the inner membrane of the mitochondria. The electronegative proteins remove the hydrogen atoms from NADH and FADH2, oxidizing the carriers to NAD+ and FAD to be used again. These atoms enter the proteins full of hydrogen ions. These protons are pumped out of the protein and across the inner membrane into the intermembrane space. The electrons are then transferred to the next protein by a carrier protein. The electrons move down the chain because each protein is more electronegative than the next. After the electrons have passed through all of the proteins on the chain, it reaches the most electronegative molecule in the chain, oxygen. The 2 electrons combine with 2 hydrogen ions and 1 oxygen atom to form water. As for the hydrogen ions being pumped into the intermembrane space, this process creates a high gradient of hydrogen ions outside of the inner membrane. This high concentration of hydrogens outside of the cell are then forced through an enzyme connected to the electron transport chain called ATP synthase. As these ions enter the enzyme, they cause it to rotate, creating ATP. This process of hydrogen ions creating ATP is called chemiosmosis and is the last step in cellular respiration.